Okay, so I know most of you were here in class on Wednesday, uh, going through 12.3. We made it through all the way through 12.3, and we made it through the first half of 12.4 of Neosis. So I'm going to do this. I'm sorry. I'll email you the exact time frame where we left off yesterday comparing meiosis and mitosis. But for those that were not here, I'm going to do you a favor and just quickly go through 12.3 and 12.4. You're going to need active notes, and I'm going to put them in the biology folder for you. All right. So 12.3 is called Other Patterns of Inheritance. Now, these patterns of inheritance that we're going to go through do not follow Gregor Mendel's principles. With Gregor Mendel and his pea plants, we talked about dominance and recessiveness, and how dominant covers up recessiveness, and it's just nice and neat. But there are some cases where that is not followed at all. Okay, so we're going to describe some of these exceptions, and then finally talk a little bit about how the environment's role does, in a way, determine which traits or genes, basically the environment plays a role with genes. Okay, so the first pattern of inheritance that doesn't follow Gregor Mendel's rules is called incomplete dominance, which means that neither of the alleles are dominant over each other. So there's not a single dominant trait, and there's not really a recessive trait either. So the heterozygote phenotype, the one that is hybrid, the one that carries the capital letter and the lowercase letter, lies somewhere in between the two homozygous phenotypes. So I think some examples of incomplete dominance are I think they're called 4 o'clock flowers or snapdragons. But let's just say I have a red flower, and we'll denote it big R for red. And it's going to be cross-pollinated with a white flower, which is little r, and we get 100% big R, little r, or pink. And you can see that the heterozygote phenotype is in between the two parents. Here we have red and white. Well, if you mix the two together, you get pink. Now, another type of weird inheritance that doesn't follow Gregor Mendel's rules is called codominance, which means that both alleles are clearly expressed in the heterozygote. So co, meaning together, and they both dominate. So if they are both expressed, they're going to be basically both are going to be expressed. So one example is chicken feather color. I think that your book mentions. It uses a little bit more technical terms, so make sure you guys look at that paragraph. Human blood. So we have, type A and type B and type O. Type O is recessive to everything. But we also have type AB. And so type AB is actually codominant because both of those alleles are being expressed for that blood type. Okay. Human protein blood cholesterol level. I don't know. Your book mentioned this. I just put it in the notes. I'm really surprised they don't mention roan cattle. Maybe this is not an example anymore, but I'm going to include it. So roan, a roan horse or a roan cow, if you were to zoom in on their fur, you wouldn't see this color that you're seeing. You would actually see individual red hairs and individual white hairs. And far away, that's the color that you get, that you perceive. Again, if you zoom in, you would see that both hairs are expressed equally. So here we have a cow, red and white hairs that occur together. Both alleles are being expressed. This would be considered a roan cow or codominance example. This is a very simple example, but a white chicken crossed with a black chicken, you get a speckled chicken, or both colors are shown through. So you see black with white or white with black. This is the right concept, but obviously it's biologically incorrect. Platypi or platypus they're not made this way. They'll take a duck and cross it with a beaver. Panda bears are not made from black bears crossing with a polar bear. Okay, but you get the idea that both traits are being shown or appear together in the heterozygote. You also have multiple alleles, a gene that has more than 2 alleles. So human blood types, we actually have 3 alleles, IA, IB, and little I. Fur color and rabbits, we actually have 4 alleles. And you can see that they're down here. Big C is basically dominant to all the other alleles. Little CCH is for chinchilla. It's a partial defect in pigmentation. It's dominant to CH and C alleles. Then we have, little CH, that's Himalayan right here. Let's take a look at the genotype for that. So it's, color in certain parts of the body. It's dominant only to the little C allele. And then we have albino, and the only way you can be albino is 2 little C's. Okay, so multiple alleles going on here with fur color in rabbits. Now more on blood types. And, oh boy, what do I want to do here? I think I'm going to try typing. Okay, so in your notes it says type A. Okay, so that means that you can have a few different genotypes. And you could be capital I, and then I need you to put it in subscript, the little a at the top, IA. This would represent a homozygous, 100% purebred type A. But you could also be IA, little I. And this means you'd be a hybrid. Now, this little I stands for the type O. So you can see up here, like I just wish they did capital I's. So it's capital I, A, capital IA with a little I. I think on one of your homework assignments, I would also accept, so if you were IA, I would accept AA. Or, in this case, this would be IO. Okay. All right. Type B, you can be IB, or IBI, that would be the heterozygote. So you carry the allele for an O. But the lazy version, I would accept BB, or for the heterozygote, BO. Okay, type AB is IA, IB, or AB, and that's it. That's the only way you can be AB. So you carry the allele for A and B, and this is where it's codominant, but it's also multiple alleles. And then finally we have type O, and the only way you can be type O is 2 little i's. Okay, polygenic traits. Poly means many, and genic means genes, so many genes control one trait. And so you typically see a wide variety of phenotypes because of that. So eye color, you can see we have lots of different examples of eye color here. Coat color in dogs, human height, skin color. So those are all examples of polygenic traits. We also have some non-Mendelian inheritance, like maternal inheritance, how it depends on the mother. So I think we've talked a little bit about this when we were talking about the mitochondria. So you get your mitochondria from your mom, and your mom got her mitochondria from her mom, and so on and so forth. Your dad got his mitochondria from his mom, who got it from her mom. So basically, if you have siblings and you all share the same mother, you guys all have the same mitochondria because you got it from that mom. The next one's called genetic imprinting, which means chemical modifications of certain genes. So different genes can be turned on and off in the sex cells, as you can see in this picture right here. And in the sperm, all the imprints could be erased and rewritten. And in the eggs, imprints could be, erased and rewritten. And how they get rewritten could be, maybe substances that they expose themselves to or stress. So I'm going to show you an example about this really cool website. It's called Utah Genetics, and we've been there before. So I'm just going to get there. Sorry for all my windows. It's called Lick Your Rats. So it says, some other rats spend a lot of time licking, grooming, and nursing their pups. Others seem to ignore their pups. Highly nurtured rat pups tend to grow up to be calm adults, while rat pups who receive little nurturing tend to grow up to be anxious. It turns out that the difference between a calm and an anxious rat is not genetic, it's epigenetic, which means it's above genetics. The nurturing behavior of a mother rat during the first week of life shapes her pup's epigenomes, and the epigenetic pattern that mom establishes tends to stay put even after the pups become adults. And so if I click on this video, which is like pretty low grade, but you know, here we have a mother rat and these are her pups. She's checking out the camera right now. But then she just goes and just nurtures and licks her pups and, you know, hey, I'm here. Don't worry. I gotcha, you know, all that. All right, so to show you.
Deep in the brain of a newborn rat pup, methyl molecules, green, silence the GR gene. When it's active, the GR gene produces a protein that helps the body relax after a stressful event. The type of care a pup receives from its mother during the first week of life can change the expression of this gene.
Okay, so you can see that this is supposed to represent like a gene. And you can see that we have low activity because all of these histones, okay, we talked about nucleosomes and histones and, you know, DNA. It's really tight and wound up. And proteins can't get in here and read that gene. I know we haven't really discussed transcription and translation yet, and we will. But it's all like locked up in here. And so if you want to turn this turn this gene on, you have to spread it out. Basically uncoil it, or it's like maybe this is like a slinky pushed in close together. And what we want to do is open up that slinky and pull it apart a little bit so things can interact with it. So I'm gonna do a demonstration with this mom. This is a pup here. I'm going to be the mom pup. So the first time I do this, I'm going to be a highly nurturing mother. All right? So enjoy. Here we go.
Wow, you're an attentive rat mother. It's surprising your pup has any fur left on its body. Your licking and grooming has seriously activated your pup's GR gene. Your pup will have an easy time relaxing after stress. Your pup's GR gene will most likely look like this for the rest of its life. In fact, the amount of nurturing you gave to your pup will have a major impact on its adult personality.
Okay, so you can see that because I nurtured my pup here, this gene is very high in activity, and as a result, it's going to be able to relax after stress. So now I'm going to hit this refresh button, and we're going to have to listen to her voice again. I'll quickly just fly through it here and we'll start it. All right, here we go. So I'm going to be an inattentive mom. Yeah, okay, here we go. And there's a piece of cheese over here. Okay, hi.
It looks like you had better things to do, since you didn't lick and groom your pup very much. Its GR gene is still inactive. Your pup will have a hard time relaxing after stress. Your pup's GR gene will most likely look like this for the rest of its life. In fact, the amount of nurturing you gave to your pup will have a major impact on its adult personality.
Okay. Now, the purpose, they're looking at anxious behavior here. And I'm not saying anxious behavior is a bad thing. Okay, it can actually be a very, very good thing. We tend to think of it like right here as this first sentence as a disadvantage, but it could be an advantage if the tables are turned, if the environment is very dangerous. An anxious behavior would be very beneficial. Okay. All right. Okay. Genes in the environment. So, your characteristics, organisms' characteristics that you see, they're not 100% determined by genes that you get from mom and dad. The environment does play a role and can affect the gene expression, and as a result, determine the traits that develop from it. So I have your book lists some examples, and I've added a couple here. You have temperature. So an example of temperature is down here. You can see that we have two rabbits, one that has pigmentation and one that does not. And this rabbit, this pigmentation indicates that it was raised in an environment that was 20 degrees or less. Siamese cats are very similar. I think it's a different temperature. I'm not quite sure on that. Don't quote me on it. But if you see a very, very dark Siamese cat, you can assume that that cat has been in cold environments. Maybe it's a barn cat. I grew up with a bunch of barn cats, and most of them were Siamese. And they were very, very, very dark in color because, we kept them outdoors, but they had the barn. But yeah, now the rabbit over here, hardly any pigmentation, so you can assume that it was in a warmer environment in which it was raised. So we did have a few Siamese cats inside of our house, and yeah, they were lighter in color, so temperature does play a role in traits. Excuse me. Your book mentions two species of moths or butterflies. They're the same species, but you can see because of different temperatures, they look different. They look like they're different animals, but really they are the same. Nutrition and stress kind of ties into a little bit of us. If you have really, really tall parents, and I shouldn't use height, but nutrition does play a role in whether genes, get expressed the way that they should be expressed, as well as stress. And I kind of wanted to go back to genetic imprinting. So we went to this website and looked at rat pups. They've actually looked at epigenetics in humans. And there was a case study with this Quebec ice storm a little bit over 10 years ago. And there was a bunch of pregnant women in this town, and they didn't have power for a very, very long period of time, which obviously is going to increase their stress level. And so they decided to follow these pregnant women to their to term, and look at their children after a few, couple of years, and so on and so forth, and basically just follow these children. And they noticed that these children that were born after the Quebec ice storm, if their mother was pregnant during that time, did have higher levels of like anxiety and ADHD and a few other symptoms, that were, they just had higher rates of them. So yeah, I'm pregnant right now, and what I expose myself to, yeah, it might play a role in my child's, genome. So okay, so that's 12.3. Moving on to 12.4, which is meiosis. Okay, so we're going to look at sets of genes that are found in adult organisms, specifically humans, and then describe the events of meiosis, break it down into its 8 phases. I know it's a lot, but they're not that hard. And then we didn't get to this today, identifying the differences between meiosis and mitosis and comparing them, and then explain how two alleles from different genes can be inherited together, basically just looking at crossing over and gene mapping. Okay, so chromosome numbers. Strands of DNA and protein inside the nucleus that holds genes, those are chromosomes. Each species has a chromosome number, and the number doesn't mean really anything. It doesn't mean like intelligence or superiority or whatever. So like humans, you can see there, Homo sapiens, we have 46 chromosomes. Dogs have 78. Roundworms have 4. We're going to look at fruit flies in the next slide. They have 8. So chromosome numbers vary among species. So if you have a complete set of chromosomes, which we do, they're diploid cells. So most of the cells in our body are diploid. The only cells that are not diploid are our sex cells, and we find those sex cells in the testes and the ovaries. So testes in males and ovaries in females. So diploid cells, they have a full set, and we also can denote it as 2N. So if I'm looking at Drosophila, their full set is 8 chromosomes. Humans have 46 chromosomes. And, the purpose of meiosis is to maintain the correct number of chromosomes inside of us. Because if our sex cells had 46 chromosomes, so if I had, an egg cell is 46, and there's a sperm cell that's 46, and you combine them together, holy smokes, you're going to have 92 chromosomes. That's not human, okay? So we have to maintain that number. So how do I have, with Drosophila here, 8 chromosomes. Well, you get half from mom and half from dad, so it means you get 4 chromosomes from dad and 4 chromosomes from mom. So these would represent the gametes, or the sex cells that fuse and combine together to get your diploid number. So with humans, we have 46 chromosomes. You get 23 from dad and 23 from mom, and boom, 46 chromosomes. So sperm, egg, meat, zygote. We also denote haploid as N. Okay, I haven't the G set turned on here, but it's down here. So haploid means a single set of chromosomes or half the number. So diploid's full set, 2N. Haploid's half N, or single set. Now, I'm going to introduce this term, I think, on the next couple slides here, homologous. So chromosomes with the same genes, one originally from each of the organism's parents. So you have 23 chromosomes from dad and 23 chromosomes from mom. That means chromosome 1 from dad, when it pairs up with chromosome 1 from mom, okay, now they're going to be homologous. Chromosome 2 from dad pairs up with chromosome 2 from mom. Now they're going to be homologous. Okay, so that's what it means by homologous. Okay, so I want to quickly review mitosis because I'm going to be comparing meiosis and mitosis throughout the phases here. So when does mitosis occur? Hopefully you're thinking about mitosis, like, oh, we talked about when a cell gets too big, it will split so it can maintain the surface area volume ratio. Yeah, that's one reason why cells divide, and they go through mitosis to do that. But another reason why cells divide is to repair. So you get a paper cut, okay? Well, those cells are going to repair through mitosis. They're going to form daughter cells that are exactly alike each other. Another example of mitosis is when you go through growth and development. So, your growth plate makes more cells, and you get taller. That's all mitosis. So those body cells, they're making more of themselves, and they are identical to each other. So we have 4 phases of mitosis, and I'll quickly review them here. So this first phase is shown prophase. So this phase, they're lined up in the middle. That's metaphase. The chromosomes get pulled apart into chromatids. That's anaphase. And then we have telophase and almost cytokinesis occurrence. So 2 do nuclei. You can see that the chromosomes start to unravel a little bit here. That's telophase. So phases of meiosis, the number of chromosomes per cell is going to be cut in half through the separation of homologous chromosomes in diploid cells. Basically, it's just saying we're going to take our diploid cell and chop it up. Okay, I shouldn't say chop, but divide it into 4 haploid cells. So we're going to go through meiosis 1, where you can see it's prophase, metaphase, anaphase, telophase, and cytokinesis. And then we're going to enter meiosis 2, prophase, metaphase, anaphase, telekinesis. But our end result is going to be with 4 cells instead of 2. So in meiosis 1, very important step that occurs is called DNA replication. Okay, so DNA replication is going to occur in interphase, and then it's going to jump into prophase 1, where our replicated chromosome is going to pair with its corresponding homologous chromosome. So that means chromosome 1 from dad's going to match up with chromosome 1 from mom. And this pairing is going to be called a tetrad. Okay, and it's called a tetrad. Tetra means 4 because it has 4 chromatids. So I'm going to get a marker here. I didn't do this today, but if purple is dad, all right, it's going to go through replication. So it's going to form another half for it. And we'll use, I'm going to change my color here, we'll use red for mom. Sorry for the color blindness, people. All right, so here's mom. Mom is going to go through replication and interphase. That's supposed to represent my centromere to hold it together. And then when they pair up, that's a tetrad. Okay. Now, another thing that occurs in prophase is crossing over. And crossing over means these 2 legs are going to swap places. They're going to wrap each other around and then rip parts from each other. They're going to hold hands, chop off their hands, and sew that back on together. Okay, I know that's really graphic. So hopefully in the next slide we'll clear some things up here. So review some parts here before we dive into crossing over. So this is a cell, okay, and this is supposed to represent the cell membrane. We have 2 centrioles, okay, they form these spindle fibers. It's supposed to represent a chromosome. And the thing that holds the chromosome together is this centromere. So here we have 2, I should say these are really chromatids. They go through replication, and that's kind of what I just drew on the previous slide. And now these 2 are sister chromatids. These two are sister chromatids, but they are still chromosomes. They are still chromosomes. And because they're paired together in a tetrad, we have homologous chromosomes. Now here's crossing over. This is what I mean by like ripping off each other's arms and sewing it back on together. So they cross over, and you can see now the end result. I don't have a 100% red chromosome. I have red with a little bit of yellow, and here I have yellow with a little bit of red. And so why is this important? Well, this is going to increase genetic variation. This is why when sex cells fuse together, the offspring don't look exactly alike. You don't look exactly like your siblings. Okay, and it's because of crossing over. This increases genetic variation in us. Okay, and if you really want to know like what's the probability, I think it's like 2 to the 23rd power. It's a lot. All right. So after prophase 1, we enter metaphase. And metaphase, if you remember, means like something about the middle. Now these homologous chromosomes are going to line up across the center of the cell. And the thing I want to emphasize is that they are not exactly, whoops, I thought I had a marker. They are not exactly on the line. They are straddling. I can't do a dash. I'm really struggling here. They are not exactly on the line. You can see that one is on one side and one is on the other, and that's what I mean by straddle. Okay, they're straddling the line. So how is metaphase 1 similar to metaphase of mitosis? I sure hope you just say, well, chromosomes are still like lined up somewhere on the center. Okay. Anaphase 1, spindle fibers pull each homologous chromosome pair opposite ends of the cell. So now we have our homologous pairs, okay, our tetrad, and they're being pulled apart from each other, which is really sad. So they're going to wave goodbye to each other and be pulled to opposite ends. Now, in telophase 1, a nuclear membrane forms around each cluster of chromosomes. No big deal there. And then we have cytokinesis, which means the cytoplasm pinches, producing two daughter cells. Now, the thing I want to emphasize is that they're not identical. They're haploid, okay? All right. How do anaphase 1 and telophase 1 of meiosis differ from anaphase and telophase of mitosis? So let's just look at anaphase 1 first. Here's anaphase. Okay. Anaphase 1, I should say, of meiosis. And you can see that we had homologous chromosomes and now they're being pulled apart. They're still chromosomes. Now, if you remember when we did our gummy lab, when we pulled them apart, they looked something like this. So they weren't full chromosomes, they were chromatids. So that's a difference right there. So in mitosis, the chromosome gets pulled apart into sister chromatids. And here, the homologous chromosomes, they just get pulled apart to form chromosomes. Okay, now telophase 1 versus telophase of mitosis, you can just say that, here we still have like chromosomes, okay? Like, they're not like chromatids. Okay, so after telophase 1 and cytokinesis, now we're going to shift gears and go into Neosis II, which is a second round of division. And the thing I want to emphasize is that there is no replication here. This will give us a good chromosomal number, half the number, okay? So our chromosomes become visible. They do not form tetrads. There's no crossing over. You're going to realize that Meiosis II is very similar to mitosis. So we enter metaphase 2. Notice the chromosomes line up in the center of the cell, just like they do in mitosis, right on that line. Then we go into anaphase 2, where sister chromatids are separated, and now they're just chromatids, just like in mitosis of anaphase, or anaphase of mitosis. So how does anaphase 2 differ from anaphase of mitosis 1? Well, you can just say that our sister chromatids, they are not identical, okay? How are they similar? Well, they're both separated into chromatids. Now, the thing I want to emphasize is that it's happening in both of those cells. In Meiosis II, we start with 2 cells, okay? So it's happening in both of those cells. So we have telophase 2 and cytokinesis. So the end product means you get 4 daughter cells, 1, 2, 3, 4. They have half the normal chromosome number, which means that they are haploid, and they are not identical. This is different from this cell, and this cell is different from this cell, and this cell is different from this cell, and you can see we have increased genetic variation. Now, all 4 of these cells, if you are male, will turn into a sperm cell. If you are female, only one of them will become the egg, and that's the reason for that is it will kind of absorb all the other cells. Okay. All right, summary of meiosis. So, interphase, DNA replication, prophase 1, homologous chromosomes pair up, crossing over takes place. Metaphase 1, homologous chromosomes line up, straddling the line. Anaphase 1, homologous chromosomes are pulled apart. Telophase 1 is cytokinesis. You end up with 2 cells not identical to each other. These 2 cells do not go through replication before meiosis II. There's no pairing up. There's no crossing over. So we hit prophase 1. We go into metaphase 2. Sorry, I meant to say prophase 2. Now we go into metaphase 2. They line up right on the midline here. These chromosomes get pulled apart into chromatids. And now my 2 cells will split through cytokinesis and telophase 2 forming nuclei. And now we end up with 4 haploid daughter cells that are not identical to each other at all. Okay, so this is where we did not get to. So I think in your notes you have a table, so you have to follow along. So the purpose of the process for mitosis, we talked a little bit about earlier. It's growth and repair mostly. Okay, body cells do mitosis. Purpose of meiosis, genetic diversity. I would also accept like sexual, well, I guess that kind of covers the next one, but it's to make sex cells, okay? The purpose of the process, reproduction, okay? So the type of reproduction in mitosis, for some organisms, it is asexual. But for us and for a lot of multicellular organisms, it is sexual. Number of daughter cells made at the end of mitosis is 2. But for meiosis, it's 4. How many rounds of division? Mitosis, there's just 1. It's PMAT. But for meiosis, you go through 2 rounds. You go through PMAT twice. Genetically identical or different. In mitosis, they form genetically identical daughter cells. But in meiosis, they're different. They're not identical. Contains homologous chromosomes? No. Mitosis does not. Meiosis? Yes. Yes, they do. Types of cells produced. Body cells. Okay, so skin cells, liver cells, heart cells. I can't think of anything right now. Bone cells. In meiosis, sex cells. Daughter cells compared to parent cells. So what did the daughter cells look like compared to the parent cells? So the daughter cells in mitosis are identical to each other. They're identical to each other, but they're also identical to the parent. In meiosis, they are different. Haploid or diploid? In mitosis, diploid, because they only go through 1 round of division. In meiosis, they are haploid because they will go through 2 rounds of division. Crossing over. Nope, no crossing over. Meiosis? Yes. Does interphase occur in mitosis? Yes, it does. DNA does replicate. Okay. How about for meiosis? Yes, it does. It does right before meiosis 1, so it still counts. So this is just showing mitosis again. Okay, PMAT, prophase, metaphase, anaphase, telophase. And this is meiosis, just showing, this looks like meiosis 1. And then this is showing the end result of meiosis 2. And so the thing I want to emphasize here, I think they were just saying, actually I don't know where I was going. Oh, how many chromosomes, okay, do you see here in mitosis? And you should say 4, 1, 2, 3, 4. So 2n is 4. I'm just going to write that in. And then over here, how many chromosomes do we start with? Okay, 4. Okay, but N means half. So if we look at the end result, we end up with 2. So really, our N is 2. So you see that in this example, N is 2, 2N is 4, and that would make sense because 2 times 2, holy crap, that's really bad, is 4. And I messed up on that 4. Yeah, okay. All right, second half of 12.4 is called gene linkage and gene maps. So here we go. Oh, okay, gene linkage means that sometimes genes are inherited together if they're on the same chromosome. And it was first discovered by Thomas Hunt Morgan. His last name is Morgan. Thomas Hunt Morgan, okay? And fruit flies. Remember, fruit flies have 8 chromosomes. And he noticed that there were certain genes on the fruit fly that were linked in a way that violated independent assortment. Because we talked about how, with Gregor Mendel in his piece, how independent assortment does one gene affect the inheritance of another. And, you know, we said no, like they didn't. We did those massive dihybrid crosses, and we saw different combinations that were not present in the parents, okay, like round and green. You know, round is dominant, green was recessive. Like we saw combinations that they didn't look like the parent at all. And so Thomas Hunt Morgan looked at, you know, what's going on here? Why are some of these genes linked together? Why are we violating independent assortment? And he noticed that during crossing over, some genes traveled together, and these genes were always within certain linkage groups. Okay. So you don't have these in your notes. It's kind of like a story. Okay. So Thomas Hunt Morgan, this guy right here, studied fruit flies, found that in some crosses, expected outcomes were not happening. He further did experiments to confirm that alleles located on the same chromosomes are inherited together. And so what he was looking at in fruit flies was a wild type versus a mutant. And so you can see on the bottom left-hand side, this is a wild type fly, normal. Wild type means normal. And this is a mutant. And he was looking at heterozygote wild type, VG plus V. Okay, so this plus indicates one type of allele, and this is without. And so they're looking at vestigial wings with a black body. So they're both heterozygote for these two traits. Looking at a mutant that was homozygous recessive for both traits. So you can see that the wings are really messed up, and they are fully black in color. So yeah, heterozygote wild type versus a mutant. So what's up with all those letters? I guess I could have just waited. But plus means wild type, in this case long wings. And if you don't have a plus, you're going to have short little wings. B plus means yellowish body in this case, and without the plus, it will be black. Okay, so you can see that there's no plus signs here, so black. But if these letters are too confusing, basically, we're just looking at a heterozygote dominant organism crossed with a homozygous recessive for both of those traits. Okay? So because these alleles are found on the same chromosome, a Punnett square would show them inherited together. There are 2 possible arrangements for the heterozygote in the above cross. If the dominant alleles A, B are on the same chromosome, they called it a cis arrangement. But if they were opposite from each other, so notice that they're not big letters on one side and small letters on the other, they're opposite, they called it a trans arrangement. And you really don't need to know that. It's just extra information. But this is where it kind of comes into play here. So consider a cross with a parent that has a cis arrangement, which means that the letters are on the same side. So when we make our Punnett square, okay, we foil it, and I guess we don't really have to foil it. This is a simplified Punnett square here. So we have AB here, a little AB, and then two little letters, and we fill it in. You're going to get 50% wild type and 50% mutants. That was what Thomas Hunt Morgan was expecting in his outcomes. However, that is not what he saw. Okay, So he expected 50-50. However, he observed 40-40, and then um, these two crosses, vestigial wings, which um, is that mutant? Yes. Wild for the yellow body, and a black body with wild or normal wings. Combinations that did not look exactly like the parents. And he's like, what is going on here? And so he called these weird offspring that didn't match the parents recombinants. And they are the result of crossing over during meiosis, which means that the alleles switch position. So here we have the cis arrangement, but they can cross over and swap sides. Okay, and he noticed that 10% of the time these alleles did switch. And if I look back at the data, he observed 10% of the time these weird arrangements. And so I'm going to go back to this, but how did they figure out crossing over and map units and all that? They had to look at the recombinants and the percentage of recombinants that appeared. So using this methodology, chromosomes of a fruit fly were mapped. There was only 8 of them. Each map unit represents how far apart these alleles are on the chromosome. The number is based on how often crossing over occurs. And so this means that they cross over like 13% of the time, or 31% of the time. And so that's actually how they've mapped it out, where the genes are located. Is looking at the percentage of time that those recombinants occurred, which correlates to crossing over. So Hunt concluded, back to your notes, these are in your notes. Hunt concluded that 2 things. Chromosomes are actually a group of linked genes, and that it's the chromosomes and not the genes that assort independently during meiosis. Okay, so when we talked about Gregor Mendel, we said that genes independently assort. But really, it's the chromosomes that independently assort during meiosis. All right, so this ties into gene maps because I kind of mentioned it just briefly before. So Alfred Sturtevant was a student of Thomas Hunt Morgan who kind of carried on his work a little bit, and he hypothesized that frequency of crossovers were related to distance between the genes. Percentage of times that the phenotypes did not appear together represent the frequency of crossovers between those chromosomes. Basically, he hypothesized that if the genes were really far apart, crossing over would most likely occur between these 2 genes, and if they were close together, crossing over between those 2 genes is less likely, which just kind of makes sense. So genetic maps, genes are shown in a relative order. Distance from each other are based on pedigree results or some other studies. The chance of chromosome breaking between A and C is higher than between A and B because A and B are closer together. Similarly, the chance of chromosome breaking between E and F is higher than F and G because F and G are closer. So basically, the closer the 2 genes are, the more likely they are to be inherited together. So, yeah. So from that, we can actually construct a map unit. Now, I think on your homework, you just have to make one. I'm not sure, so make sure you refer back to your notes on this one. So one map unit is 1 crossover for 100 offspring. Basically, it's 1% out of 100. So here, if we have genes B, CN, and VG, and they give you these percentages, basically you just have to arrange the genes in a way that makes sense. So if they said that B and VG are 17 map units apart, well then you would make a line and you say, okay, well, all right, I'll stick B here, just like they did on this diagram, and VG, and I just know that they're 17 apart from each other. Okay, so 17. Whoa. Okay, and then if they gave you some more information like B and CN, it's 9% from each other. So that means it could be here or here. We were like, well, I'm not really confident, putting it here or here. So if I read the next bit of information, they say CN to VG, well, now I have a relative point where I can pinpoint where CN is exactly. And they give you that CN is 9.5. Now, if CN was over here, and that was supposed to represent 9 from CN to B, but then I have 9.5 from CN to VG. You'd be like, nope, that's incorrect. There's no way CN could be there. So CN would have to be here to make all the data line up to each other. So we'll do a little example down here where it says B and A are 6%. Sorry, CO means crossover. They cross over 6% of the time. So that means B and A, so I don't care where I'm going to put. I'm just going to put B here. All right, so here's B. And A, so it's like 6. It looks like I got some big numbers here. So I'll just put A right here. And we'll just see how it goes. You might have to erase, who knows. And that is supposed to represent 6. Okay? So I'm going to write 6. Okay. I read the next tidbit of information. Gene A and gene C cross over 12.5% of the time. All right. So that means I could put C here, or I could put C like approximately here. Okay, so I don't feel confident yet. Let's read a little bit more. Oh, good. They gave me information about the relationship between B and C. So gene B and gene C cross over 18% of the time. So if I were to put C here, is that 18? You know, there's no, okay? Because this is like, well, from C to A, it's 12.5, and then that's supposed to be 18.5. No, it'd be like half that, a little bit less than half that. So C does not go there, okay? So C would fit better here because, you know, this would represent the 12.5. And then if you add up the distance between C and B, 12.5 plus 6, oh my gosh, look, is 18.5. So that's the correct order, B, A, C. Okay, they're not that hard. So I think the ones they give you in the book are simple. So this is an example just showing that, yeah, they've actually have mapped the genes out. Okay. So question for you, where in the chromosome is the purple eye gene located? All right, hopefully you look, 54.5. That's it. All right, so that wraps up chapter 12. So you are going to, you have to do 3 assignments, okay? I'm going to type them up here for you. There are 3 assignments. There's a 12.3 worksheet. Well, I guess I could have just abbreviated. It deals with the other patterns of inheritance. Okay, so that's codominance, incomplete dominance, multiple alleles, polygenic. Okay, then there's a 12.4 meiosis worksheet, and it's just meiosis. I mean, it's just meiosis. I thought about combining these two. Like, I thought I was going to be here, but I'm not. So I'm sorry. I'm just throwing you this all this information on you, but the worksheets are still separate. So it's just about meiosis. So then move on to just meiosis, and just, you know, use your book because you're going to have to sketch some phases of meiosis. All right, and then there's another worksheet. It's 12.4, and it's just about gene linkage and mapping. I can't even type. So the stuff I just covered with Thomas Hunt Morgan, and it's just a one-sided worksheet. It's not difficult, okay? So those are the 3 things. So get started on it. Start with 12.3. You also have Friday. Okay, but Friday is your last day. Please, 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 please be productive. Okay, if you are struggling, ask to go to Mrs. Deal if you are one of her students. Okay, I will email her a key with page numbers to help you. Jessica is not here this week, and I am not feeling great. So I do apologize. This is not ideal. So on Monday, we'll do like a massive classroom review. And we're probably just looking at a test on Tuesday, okay? So thanks, and again, I'm sorry. Email me if you have questions. I have my laptop with me.